This is podcast number 36. And eventually we're going to talk about all the major molecules except for nucleic acids in food. So we'll talk about carbohydrates, talk about lipids, we'll talk about proteins, and how convert the energy in the bonds of those molecules that you eat into energy that you can use easily, and that'll be mostly ATP. For this first one, we're going to talk about mostly carbohydrates. So if we think about mammals drink milk as soon as they're born, and it has complete nutrition for a newborn. And the components of milk and digestion co-evolve, so evolve along the same pathway, in that as the components of milk changed and were subject to natural selection to improve survival and fitness of offspring, those offspring's ability to use those macromolecules in milk also evolved. One of the components of milk is lactose. Lactose is milk sugar. It's a disaccharide, so it's two monosaccharides, two carbohydrate units hooked together for a total of 12 carbons. And it's a combination of glucose here and galactose there. 10% of all milk sugar is lactose sugar, and we're going to talk about one of the components of lactose. Lactose is broken down into galactose and glucose. Sorry, I had those backwards on the last slide. Doesn't really matter. They're both 6-carbon sugars. And if you see in this slide, this is a cell that lines your gut, has increased surface area with these finger-like projections called microvilli, and you can see a lot of little dots here. It sort of looks like dirt on the micrograph, but those are actually dots that are labeled with antibodies, and this antibody is detecting an enzyme called lactase, which breaks this bond between galactose and glucose and allows you to use milk sugar. In people who are lactose intolerant, they have much lower concentration of this enzyme at their brush border in their microvilli cell, in the cells that line the gut. And you can lose expression of lactase over time, from lots of expression when you're younger to maybe less expression when you're older. You become more lactose intolerant as you get older. So you're going to see a ton of detail about glycolysis, the process of breaking down glucose, in your book. That's very overwhelming, I think. We're going to use a handout to emphasize what I want you to know about glycolysis. And I'm going to switch to that now so you can see it. This is posted on Brightspace under handouts. And you're going to see this pathway, if you stay within biology, over and over and over again. You'll see it here, you'll see it in cell and molecular biology, you'll see it in biochem, in advanced biochem, and then if you continue on to med school, you'll see it there too. So why? Why do we obsess on this pathway? So it's a really good example of a biochemical pathway that takes energy in the form of covalent bonds between atoms in a compound and converts that into an energy form that your cells can use. And it's really well studied, and it turns out it's really important in cancer. So you're going to see a lot about this pathway. I'm not going to make you learn all there is to learn about this pathway. Just a few things. So the underlying molecule names, I want you to know those. And then there's one enzyme that's not on here that's on the slides when we go back to the PowerPoint that I want you to know. Okay? So you're going to start off with a 6-carbon molecule, glucose. I don't care that you can draw that. I just want you to know that it's a 6-carbon molecule, and glucose is one of the 6-carbon molecules in lactose, like in milk sugar. And this 6-carbon molecule is going to get phosphorylated into glucose 6-phosphate. So the 6 carbons are numbered 1 through 6, and glucose carbon number 6 gets a phosphate attached to it, and that's now called glucose 6-phosphate. This phosphate comes from ATP, takes the terminal phosphate off of ATP, becomes ADP plus P, and that P goes on to glucose. Then those 6-carbon molecules rearrange into a different shape. Still is a 6-carbon molecule, but now is in a different shape, and it's called fructose instead of glucose. Still with a phosphate on number 6. And then you split another ATP and into ADP and P, and you put that phosphate on now carbon number 1. So this molecule is called fructose 1,6-bis. Bis means 2 phosphates. So fructose with 2 phosphates on it. Now, what have we done so far? Not a whole lot. So we have a 6-carbon molecule here. This is also a 6-carbon molecule. Now it's got 2 phosphates attached to it. And this is the thing we're trying to make, ATP. But instead we have spent 2 ATPs. Okay? So you can think of this as like a loan to get your small business started. You think of ATP as cash. It costs you $2 to get started, but you're going to make some money at the end. So this 6-carbon molecule is now going to split into 2 3-carbon molecules, and I draw them both on each side of the page. Okay, that's just to show you that the same thing is happening twice. Okay, and this is cut off by the recording, but this molecule is glyceraldehyde 3-phosphate. So it's a 3-carbon molecule called glyceraldehyde, and it has a phosphate on carbon number 3. So this 6-carbon molecule splits down the middle. 3 carbons and a phosphate go in one direction. 3 carbons and a phosphate go in another direction. Both of those molecules are called glyceraldehyde 3-phosphate. Okay? Now, in glyceraldehyde 3-phosphate becomes 1,3-bis phosphoglycerate. So carbon number 1, carbon number 3, both get a phosphate on them. And because it's 2 phosphates, we're going to say bis for 2, phosphoglycerate. So glyceraldehyde 3-phosphate becomes 1,3-bis phosphoglycerate. Okay? Those 2 molecules there, in the conversion of glyceraldehyde 3-phosphate to 1,3-bis phosphoglycerate, an electron carrier called nicotinamide adenine dinucleotide, NAD, gets reduced. So it was NAD+. It gets an electron attached to a hydrogen added to it. Since I'm adding electrons to it, it's reduced, and it becomes NADH. So this is an electron carrier. If we were face to face, I would show you a battery. You can think about it like charging up a battery. Later on, in another podcast, you're going to see that the batteries that we charged up, we're going to discharge later and turn them into ATP. So we're going to use this energy, and this is the first spot where you get energy out. And you got 2 charged up NADs, 2 reduced NADs becoming NADH, because this molecule is split in 2, and everything is happening twice. below this split. Okay? Now we have our 1,3-bisphosphoglycerate here and here, and we're going to take that now for the first time we're going to actually make ATP. So let me back up just a second here. So glyceraldehyde 3-phosphate going to 1,3-bisphosphoglycerate. We reduced an NAD, made NADH. We also added an extra phosphate onto this molecule. Where did that phosphate come from? In the past, it came from ATP. This phosphate is just hanging around in the cytoplasm. Remember, in a previous podcast, I told you that I go through about 500 pounds of ATP in a day. When you're splitting that much ATP into ADP, there's a lot of extra phosphate around. And so you just grab one of those extra phosphates and add it on to here to get 1,3-bisphosphoglycerate. So this 2-phosphate 3-carbon molecule then takes one of the phosphates off of here and adds it back onto ADP to make ATP, and is left with a 3-phosphoglycerate. Okay, not the same as this molecule. It's rearranged, even though they look the same. It's 3 carbons. It's got a phosphate on them. They have the different shape, so it's a different molecule. 3-phosphoglycerate versus glyceraldehyde 3-phosphate. Okay, and we made 2 ATPs. One here, one here. Okay, up here we spent 2 ATPs. One here, one here. Okay, and now we're even Steven. Okay, borrowed $2. And then got the $2 back. Plus I have 2 reduced NADH molecules that I will use later. 3-phosphoglycerate, that phosphate moves to the number 2 carbon, comes 2-phosphoglycerate, and then that 3-carbon molecule gets rearranged into what's called phosphoenol pyruvate. Okay? And then in the final step, that 3-carbon molecule goes to, with a phosphate on it, take that last phosphate off, add it to ADP, and make ATP. And again, over here. And you end up with pyruvate, also called pyruvic acid. They are the same thing in terms of this class. We're going to call them the same thing. And it's a 3-carbon molecule. So at the top, I had a 6-carbon molecule. I ended up with 2 3-carbon molecules. And by gradually oxidizing this molecule from here down to here, this is an oxidation process. Each molecule becomes more and more oxidized. You're stripping electrons off of that molecule. Then I'm going to make 4 ATPs total. 1, 2, 3, 4. But I spent 2 at the beginning to get this going, minus 2. So I have, even though I made 4 ATPs, I had 2 ATPs net gain. But I also got 2 reduced NADs. NADH is reduced NAD. And I'm going to use those in stage 2, which we'll talk about in another podcast. Okay, so that's the handout. Those are the things I want you to know. Notice there aren't any enzyme names on here, but when we go back to the slides, I'm going to add 1 enzyme name. Okay? All right. So this is just your book's version of what I just showed you on that handout. So glucose, 6 carbons. Add ATP. Add. Now, in your book, talks much more about the enzymes. Okay? This one I'm not so worried about. Okay? But you can see here I added a phosphate onto the number 6 carbon on glucose, glucose 6-phosphate. And then I added a phosphate onto the number 1 carbon, glucose 1,6-bisphosphate. Okay? And then I split them. Well, sorry. And then I split them into glyceraldehyde 3-phosphate. Okay? That's this 3-carbon molecule here. These are, every time there's an arrow on the handout or on these slides, that's an enzyme. Okay, we're not going to worry about the enzymes with one exception that we're going to, this guy here. This says FOFO, it should say phosphofructokinase. We're going to talk more about that guy. Okay, PFK for short, phosphofructokinase, with an S there. Then those two 3-carbon molecules, they're showing them side by side here on the handout. I showed them on different sides of this, parts of the slide. Okay. You can see that we went from 3-phosphoglycerate to 1,3 bis phosphoglycerate, and then we take off, and that's where we charged up the 2 NADs to make NADH. Okay, then we take off the phosphates on each one of those to make ATP, and then do it again and end up with 2 pyruvates. Again, if we go back to our handout here, we went from glyceraldehyde 3-phosphate, 2 of those, 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phospholipopyruvate, and end up with pyruvate. They're skipping a couple of those steps on these slides. Okay. Harvest E, harvest energy. Okay, you're harvesting energy in the form of reduced NADH, harvesting energy in the form of ATP, harvesting energy again in the form of ATP, and you end up with 2 pyruvate molecules. These pyruvates are more oxidized than the glucose I started with. From the top to the bottom is an oxidation. Each one of these is an oxidation reaction. Okay? And then those 2 carbons you're going to see in the next podcast are going to be reduced even, or oxidized even further to get even more ATP out of that. So at the end of glycolysis, you end up with 2 ATPs net gain. You made 4 total, but you took 2 to get started. 2 ATPs net gain. And glycolysis happens in the cytoplasm. Okay? And you will notice, hopefully, that there's no oxygen anywhere in this glycolysis reaction. Oxygen is not required for glycolysis. So when cells run out of oxygen, which they need in order to make more ATP, they can always do glycolysis, which does not require oxygen. And you only get 2 measly ATPs out of it, but you can still do it if there's no oxygen around. Okay, and get some energy out of it. And this is why glycolysis is important for the study of cancer, is often cancer cells grow much faster than their blood supply, and so oxygen is not necessarily around, and they can always do glycolysis. And cancer cells make special versions of some enzymes that allow them to do glycolysis really fast. Okay? So 2 NADHs, 2 ATPs, 2 ATPs again made, but you started with 2 ATPs that you used, so 2 ATP net gains. Okay, and this is an oxidation reaction from the top to the bottom. All right, so we said we're going to talk about this again, and in this slide they spell it correctly, phosphofructokinase, or PFK for short. When scientists were looking at how things built up at different parts of this pathway. So they measured the concentration of all those intermediates that we talked about. Glucose 1,6-phosphate, fructose 1,6-phosphate, 3-phosphoglycerate, all that stuff. And they said, okay, how much of each one of these things do we have? I found out that there's a big buildup of the second substrate, so glucose 6-phosphate, right before phosphofructokinase. And all the other substrates had relatively low concentrations of things. And how they interpret that data was that this enzyme, the first enzyme that adds the phosphate onto glucose, glucose 6-phosphate, that works pretty fast. But then that thing really builds up a bunch, because the next enzyme that goes to glucose 1,6-phosphate works really, really slow. And that's the enzyme we're talking about, PFK. And because it worked, and all the other guys work really, really slow. Because all the other guys work slow and work fast, and this guy works slow, if the cell were going to regulate this pathway, this is the slowest part of the pathway. Okay, when you're hiking, a good practice is you put the slowest hiker at the top of the pack of hikers, and that makes everybody hike as slow as the slowest hiker, so that you all stay together. So this is the slowest hiker in this pathway, and if I make this guy faster, the whole thing gets faster. If I make this guy slower, the whole thing goes slower. But if I regulate this one here, it's still going to back up here. And these are the concentrations of the different things that they measured. And this is the concentration of the enzymes for all the things they measured, how much enzyme was in the cell, and then here's the concentration of the cell substrate building up at each spot. And the S2 1500 is way higher than anything else in this pathway, so things are really building up right there before phosphofructokinase. So they thought that's probably the thing that regulates glycolysis. That's what it tells you, is it's backing up. Okay. So we started talking about homeostasis and how do you regulate glycolysis. You got to know if there's glucose around to break down, I'm going to start breaking it down. And if there's no glucose around, I don't want to start breaking it down. Or if I don't need any energy, why would I break down my glucose? So those are things I can do to regulate this pathway. Okay? Enzyme 1 produces intermediate 2. Enzyme 2 produces intermediate 3 in the pathway. Okay? And we talked about the different intermediates in the pathway. Okay? And that enzyme, PFK, is slow. Okay? And most of the other enzymes are fast. So what sort of things regulate PFK that are in the cell? Okay? So they give PFK its normal substrate, and it works pretty fast. Okay? They gave PFK citrate, which is something you'll see that happens in stage 3. It's one of the components of stage 3, and it goes pretty slow. If they give it citrate but then switch to AMP, okay, after a bit, after 7 minutes into the experiment, it starts to go faster. Okay, and this is telling them, okay, PFK increases with AMP. Well, AMP is a breakdown product of ATP. So if you're breaking down your ATP and you don't have much left, AMP may be one of the things that's around. So AMP binds to PFK and says, hey, crank it up. Let's process more of that glucose so we can make more ATP. And that's one way you can upregulate how much glycolysis is happening, by adding one of the breakdown products of using energy, AMP, from ATP to ADP to AMP. You build up that AMP saying, okay, I need to make more ATP. That binds to the PFK and it turns on the glycolytic pathway. Okay? So let's look at ATP. Well, what's its effect on PFK? Here is the activity of PFK, and here's ATP. When ATP is low, very low, it has a stimulatory effect on PFK. So again, if you are running low on ATP, maybe you've converted most of your ATP to ADP and then AMP, which also turns on PFK, that will turn on PFK if ATP is low. But if you've got a bunch of ATP out here on this axis, okay, much more higher concentrations of ATP, it actually shuts down PFK. So it'll go back and bind to PFK and say, there's no need to break down any more glucose because I've already got lots of ATP. And so it'll cause PFK to work even slower and build up glycolysis at the point of PFK. And then as you use up that ATP and you get back to low concentration ATP, PFK will turn back on. Okay? It slows down with high concentrations of ATP because that's adaptive, and that you don't want to keep breaking down glucose if you already have lots of ATP, because the purpose of converting the energy in glucose is to make ATP. So if you already have a bunch of ATP, you don't need to break down glucose. All right, we're going to continue with these ideas in the next podcast.